Hello and welcome to the first day of MD and DI's Focus on Fundamentals course, Keeping Your Biocompatibility Program Current in a Changing Regulatory Landscape, sponsored by Nelson Laboratories. I'm Chris Keach and I'll be your moderator today. We have just a few brief announcements before we begin. First, this webinar is designed to be interactive. The dock of widgets at the bottom of your screen will allow you to learn more about today's speaker, download resources, share this webinar via social media outlets, and participate in our Q&A session that takes place at the end of our presentation. Please note that we'll try to get to as many questions as we can in the time that we have left at the end of today's program, but if we're not able to get to your question today, someone will get back to you after the program is over. Also, the slides will advance automatically throughout the event. You may download a copy of the slides via the Resources widget. And towards the end of our webinar, we will ask you to complete our survey found on the right-hand side of your screen. Please take a moment to fill this out before leaving us today, as your feedback will provide us with valuable information on how we can improve future events. And lastly, if you're experiencing any technical problems, please click on the Help widget found at the bottom of your screen or type your issue into the Q&A text area, and we will be glad to offer one-on-one assistance. And now on to today's class. What is changing in the global regulatory landscape? Discussing today's topic from Nelson Laboratories is Thor Rollins, Director, Toxicology and E&L. To learn more about our speaker, please visit the Bios widget. It's with great pleasure I now turn this special session over to Thor to begin. Thor, take it away.
Thank you, Chris, and I want to tell everyone that I'm super excited to be here today to talk about what is changing with the global regulatory landscape, specifically what's changing with standards, regulations, mostly both here in the United States and in Europe, but also impacting ISO 10993, which would impact the global community at large. I have to kind of start with a little bit of information. So this slide deck's a little bit out of date because just in the last 2 weeks, there's been some significant changes to what I'm going to be talking about. Specifically, the new ISO 10993-1 was actually published, so you can now purchase that on the ISO website. And we just received the latest 10993-18 draft this morning. So some of the slides we'll still talk through these documents and what's happening and what's changing with them. But some of the slides may have a little bit of outdated information, but that's what you get right now with biocompatibility, is that if you haven't heard what's happening in the last 2 weeks, you're probably out of date. So good timing for this. I also want to let you know kind of the stages of the class in the next 3 days. My focus is solely going to be on the standards and the regulations, the ISO 10993, the MDRs in Europe that's coming out and really having an impact, and kind of what's happening in those standards to help what's next, what's the next kind of impact to the medical device market. Then tomorrow we're going to be talking about more in detail about the chemistry impact. Specifically, we'll talk about that new ISO 10993-18 draft that was released to the ISO members. What's in there, what kind of impact that's going to have potentially going forward, and how we're starting to see the FDA specifically take this chemical characterization and apply it to medical devices. And then the last day, we're going to kind of talk about what approach, how do we wrap up all of this into an actual biocompatibility approach. These changes, this new way of doing chemistry and risk-based approach, how do we wrap it all up? So that's kind of the 3 days that we're going to be talking about, but I'm really just going to kind of go through the standards and talk about what's changing and what we're seeing from the ISO 10993 documents and also about the new MDRs. So first off, I think it's important to really kind of talk about what biocompatibility is and how does the FDA approach it today versus what they've done previously. So biocompatibility really is, and I think this is very important to understand, is that biocompatibility really is just trying to understand how compatible our materials and the processing that we're doing to those materials are to the human body. And it should be a duh, I think, for most of us. But when a lot of us end up doing a biocompatibility assessment for materials, we rely so much on, okay, what testing do I have to do? And you know, do I just kind of checkbox those tests and not so much on really understanding what biocompatibility means and understanding, okay, what are the risks of my device and how it's used, the unique risks of that device, that material, what I'm doing to those materials, and how can I mitigate those risks? What steps do I need to do, not just to show to the FDA that it's safe, or to a notified body that it's safe, but to your company, to yourself, because ultimately you're putting this into somebody's mother, somebody's daughter, and so that should be a very important part to your evaluation of a medical device. Along with that, you have to think about failures with this big picture approach. How can a device fail in use, and does that increase the risk of that device, and how do you mitigate those risks? So not just if everything goes perfectly smooth with the procedure, but what could go wrong, and not ridiculous case, right? But what a realistic worst case could be with a failure, and how do you mitigate that risk? And with that, we have to really understand the device. We have to understand what comes off that device chemically. We have to understand how the body interacts with the surface characteristics of the device. So all this becomes very important to understand the materials and the processes to really understand the risks. Then the next step in this whole big picture of biocompatibility is engineers really don't like to keep things status quo. Right, the engineers are constantly changing these devices, either out of forced changes, maybe a supplier has gone out of business, or maybe they're not making a certain material anymore, or maybe it's a bettering of that device. All these changes also need to go through the same assessments, what this change impact could have on the body, and how do you mitigate that. So really these standards that we're writing now are really now trying to understand this risks of these materials and like I said, residual processing, and trying to give guidance on how to understand these risks and how to best mitigate them. So that's the approach, because ultimately we're all about first do no harm, right? That's the objective we all have. That's what I love about this industry. Even competitors, we all put all that aside when we start looking at these documents, because ultimately what we care about is trying to make the safest devices possible for the patient, but also getting the most up-to-date technology out there as fast as possible to help impact patient life. So that's, to me, the reason why I'm in this business, the reason why I love working with these companies, because I think everybody's goal is pretty much that. So now we'll get into the actual what is changing, and like I mentioned, this link I have here, this was actually for the public draft of ISO 10993-1. Well, now that standard is, like I said, released, so you can purchase it off the ISO website. So the contents that we're going to go over is still the same, because it was in the final draft part of the standard, which is that FDIS. And really at that point, you really can't change anything technical. So everything we're going to kind of talk about is still there. It's just no longer in an FDIS stage. It's public. So kind of what we just talked about in the previous slides, the biggest changes have to do with forcing companies to take more of a risk-based approach. So I've been doing biocompatibility for 17 years, and so I've seen almost a 360 kind of approach to biocompatibility, where we've gone from more of a risk kind of assessment when I first started working in the biocompatibility realm to transitioning to a checkbox mentality, where in the last 10 years, all the companies, what they would do is that chart that is very common in the standard, they categorize their device, they just do the testing that chart has, and then they'd move on. And that was kind of what was expected from the regulatory agencies at the time, and so that was pushed down to the manufacturers, and it was easy, right? I mean, as the scientists and engineers, we love checkboxes and we love protocols and standards, and so it was easy just to go check. Okay, I feel like I'm done my part. The problem with that is there's two scenarios where that just doesn't make sense. The first is that that may make sense when you're dealing with things like stainless steel or titanium or even teak. But medical devices today are getting more and more unique types of materials. We're starting looking at nanomaterials or certain polymers that are engineered specifically for this particular medical device and its use. Or we're starting to use medical devices in unique ways, where we're getting very unique contacts with the body, where we're just not implanting it into muscle or into bone. Maybe we're going actually into the brain directly, or we're starting to look at very unique applications for some of these materials. And what we've learned when you start to really evaluate these materials and processes is that these historical animal tests that we've ran forever are just not that good of predictors outside of very simple materials. In someone may argue they're not very good predictors for even that. And so the more and more we learn about these traditional biocompatibility tests, the more we understand that they just don't really evaluate the risks like we want them to. And so what we really want, the new guidance, the new 10993, is to really understand, each company should really understand the materials. You shouldn't just purchase something from a supplier and have no idea what it is. They may even say medical grade, which means absolutely nothing, right? I mean, you really have to understand why it's classified as medical grade. It may say USB class 6 on it, but do you know what USB class 6 really means? So instead of just taking their word for safety, you have to understand yourselves what that material is. What testing does it come with? What are you doing to that material internally that could change those results of that test to really understand the risks of the portion of that device to the patient? And that's what we're trying to give guidance on, is how do you do that? Like, what kind of information is required to do this biological evaluation? And then once you have that evaluation, that leads you to what testing you have to do to fill in your gaps that you don't have that information for. And even though this requires more upfront work, it's actually better a lot of times for the manufacturer, because a lot of those biocomp tests historically are very expensive and long. And once you do a really well-done risk approach, those tests may not be needed based on the information you have. So ultimately, it benefits just not the patient because you get a better approach, but it benefits the company because a lot of times these new approaches are less burdensome from a cost and time point. Now, the old 10993, the previous version, still had this essence in it. This chart that we're showing now is actually it's currently still in the draft, or the new published version, but it was always in the standard. And this is kind of the first approach to help people understand a risk-based approach. The first question up there, does your device have intended patient contact, is actually one that I think a lot of people miss, because we see a lot of times the device submitted to have testing done either on a portion or on a device that's not intended to have patient contact. And so we really start to have our initial risk assessment is, do I need to worry about biocompatibility, is your first question. But then once you answer yes, it contacts the patient either directly or indirectly, then we really want to understand the risks, and that goes into comparing products to materials that you already have in use. So a lot of my customers that I work with, they're not just making one device. They'll have a series of devices, and a lot of times those devices are all made from materials sourced from the same vendors. And a lot of times those materials go through very similar processing. But historically, they would just checkbox the test that they would have to do, and what ends up happening is they're testing the same raw stainless steel, going through the same milling process over and over and over again because they feel like it's required from a regulatory standpoint. Well, this approach really, if you understand those materials, if you already know their biocompatibility, you already know the impact of your processing, then the burden of that risk assessment's already mostly done. You just have to be able to put something into a good format to be able to address the risks and have discussions with the FDA and your notified bodies. I do want to have some time right now just to plug pre-subs or pre-discussions with regulatory bodies. They go a long way, and I know on the third day, Audrey Turley will kind of get into this a little bit more, but man, we see so much more success with this risk-based approach when you can develop a risk assessment, develop a plan to address those risks, have a really good conversation with your notified body or the FDA. We recently had TÜV Süd in-house, TÜV, and the same discussion there is they really would like to see these plans up front, and so they're not saying no after everything's already been done. So, and I know the FDA loves that too. So a good dialogue with the agency or with your notified body on that risk assessment will help mitigate any risks to your company, right? So you won't be surprised necessarily at the end, but also allows you to get that good feedback from them as far as what they're wanting to see for your specific devices. So please look at this chart. It definitely has the concepts that you want to understand for your materials and to put into your risk assessment. So when we talk about this, the physical and chemical information, and we're going to show you the chart here in a minute, but one of the main things that's being added to the new 10993-1 is this requirement for physical and chemical information. And I want to kind of mention that we're using information, and we say physical and or chemical in the chart. And the reason why we do that is because we mentioned before, you need to understand the type of materials and what you're doing to those materials to really understand the risk of your device. We had one, and if you're listening, I'm sorry to call you out, but we had one customer of ours that actually went down to the local Home Depot or store for home materials and purchased some material to put in their medical device. And I'm not saying that, well, I'm not saying that that's, well, I am. I wouldn't do that myself personally, but it's hard to understand exactly what you have in that material if you do that approach. So understanding those materials is vital because that person was just relying on some of these animal tests to say that material is safe. And if it passes the sensitization test, then they feel confident that they can put that material in the body. And I can tell you, knowing what I know about some of these animal tests, I wouldn't feel confident if that material just passed those to be put in my body. I would want to know more about the material itself and its inherent risks and not just rely on those animal tests. So that's what we're trying to get to. And part of that physical and chemical information, I know I've mentioned it before, but it's important to stress, is not just the material, but it's what you're doing to it. I can't tell you how many times in my 17 years I have tested titanium. It would be ridiculous if I tried to tell you. So I'm not really worried about titanium or ASTM grade stainless steel. That's not my concern necessarily. What I'm concerned about is what you're doing to it. Are you leaving residual oils? Are you doing a washing step? And if you are, does that have detergents? Are you rinsing those detergents from that material? All this needs to be evaluated as your risk. So when we say physical and or chemical information, it's just not your material, it's also what you're doing to it. And I also want to stress that we don't require that you have to do chemistry on every single device. There are some devices that the risks may not warrant to do a full chemical profile. A very limited skin contacting device, for example, may not need to have a full extractable leachables done to understand its risk to the patient. Or if you're using well-known materials and processes you've used before, then you may be able to address those physical and chemical needs without going through that extensive type of testing. But if your risk is such and you don't have sufficient information, then that chemical information really helps supplement your risk assessment. The other thing that I want to kind of mention is that historically, chronic toxicity and carcinogenicity was really not addressed. So in the standard, they were mentioned, and there was X's for certain risk categories, but those tests most of the time, especially carcinogenicity, was just not done. Going forward, because we're doing a risk-based approach and because we now have more and better access to chemistry, both those endpoints should be addressed. And they should be addressed first through this chemical information. That's the best way with a toxicological assessment at the end. That's the best way to address those endpoints instead of just those tests are very long and expensive, and if we can address them through the chemistry, then that's the better way to do it. Now, talking about assessments, it's important to understand that this is where the upfront work comes in, right? So when we're talking about risk-based approach and putting it into a very good format to have dialogue with your notified body or the FDA, this expert assessors is starting to become more important. Before, anybody could do biocompatibility because it was more like a checkbox. If you could follow a chart, you could do biocompatibility. Today, it's much more of an expert. In fact, you really need someone or a group of people that have knowledge in chemistry, knowledge in biocompatibility, and knowledge in toxicology to be able to make these initial risk assessments and define a test strategy that would mitigate those risks. And so that's becoming more and more important. And to have good dialogue with your notified bodies or with your FDA, it's important to have someone that can have that dialogue, that has those backgrounds. So just keep that in mind, that is probably one of the biggest impacts we've seen recently with customers is previously they've not really needed that expertise, and now they're starting to need more. And so that's the only, I guess, downside. There's less. A lot of times there's less testing that is required based off the risk, but you do need someone that can perform that. So you need someone who can do this biological evaluation of the medical device to determine the acceptable testing plan based off of the material and processes and its intended use. Understand the adequacy of material characterization, and we'll get to this more with Dash 18 and tomorrow, but one of the big problems right now and what we're trying to solve with the new draft of 10993-18 is the vast differences between chemical characterization. Between lab to lab to lab, there really isn't much consistency with how an extractable leachable test is performed, especially with medical devices. In the pharmaceutical world, where they've been doing extractable leachables on containers for much longer than we have, it's much more well defined as far as the approach. But with medical devices, there's still a lot of ambiguity because it's so new. And you have a lot of labs trying to do this the best they can. So Dash 18 is the new draft is hoping to get more of a unified approach. But until that standard is out, and even afterwards, because there's no way you can do a protocol for this, you have to have someone who can understand the adequacy of that material characterization. What material getting back? Does it give you the right results you need? Because we're basing so much of our safety now on this chemistry. That chemistry better be rock solid. The two biggest mistakes in a chemical profile is either not identifying, missing a whole compound or misidentifying a compound, because from a toxicological standpoint, if it's not there, you can't assess it, and if it's misidentified, you're assessing the wrong thing. So it's really important to have somebody that can adequately assess the material characterization approach. In that plan, you need someone to be able to have rationale for what tests and you're doing and which ones you're waiving, how to interpret the data you have, and the need for any additional data that may be lacking through a gap. So I do want to mention in this particular part that the MDRs that are being enforced soon, this is a big part of that. So devices are being recategorized. Devices that have CE mark have to go back through their notified bodies to be evaluated per the new MDRs. And these new MDRs have very similar approach to this risk-based biocompatibility. And so with that, your notified bodies are going to be looking at all these devices that have been maybe already on the market for a very long time and assessing them to the new standard. We already have companies that are coming to us asking us for help to go through their catalog and doing kind of a gap analysis on per the new standards to understand where their gaps are, so that when they go to their to the notified bodies with this new approach, that they feel confident that they can get their devices reapproved. So just so you know, that MDR is going to have a large impact on medical devices in Europe, and it's coming closer and closer, so just make sure you're prepared for that. So this is the new chart in the new ISO 10993-1. And as you can see, the big changes to it is in this, we added a new category, which we already talked about, physical and or chemical information is required. As you've noticed, this is the only category where we have an X, right? So this is the only one that's required. Everything else is needs to be evaluated. So this is from the draft. This isn't the one from the current standard out, but it's very similar, if not identical. So you can evaluate every single other endpoint, and I think that's why it's key. We also have this draft now in an informative annex in this chart. So it's no longer in any kind of normative area, or let me help explain that, I guess. An informative annex, right, is just for information. It's not like required. So we're really trying to push this risk-based approach. So with this new table, you would provide material, physical, chemical information that's required, no matter what your device is. As you can see, it's marked everywhere. And then everything else you would evaluate. You would evaluate if that category is needed, and if not, you need testing. So as you can see down in the bottom, the X's means prerequisite information, and those E's are endpoints to be evaluated in the risk assessment. And there's some ideas on how to do that. So this is the approach that regulatory bodies are going to start enforcing now that the new standard's out. And they're already kind of have. I mean, the FDA's really already gone. If you read their drafts or their guidance document they released in 2016, it's already on a risk-based approach. So there's not much more. I think they may go more this way, but they've already pretty much set in their ways when it comes to this type of approach. Okay, so that's kind of what Dash 1 has, the new one that's just released. Like I said, go ahead and go purchase the document on the ISO, go through it. I think it's going to be worthwhile. Just make sure you do it after a lot of coffee or not early in the morning or late at night, because as we know, ISO standards may not be the easiest thing to read. Now, talking about Dash 5, so for ISO 10993-5, this is specifically for cytotoxicity, which I know a lot of people use as kind of a screening test. With that, there are a bunch of different versions now of cytotoxicity. Each one of them has kind of their own sensitivity and their own kind of unique results as far as what level you look for, if they're looking for cell death or cell different types of cell endpoints. So this is a source of frustration through the industry right now, because they may run two different types of cytotoxicity tests and get two different results. Because they could be looking at two different endpoints, or the acceptance criteria may be different, and the sensitivities may be different. So we are currently working on a round robin through a bunch of labs throughout the world to run very similar, the exact same materials controls with these tests to understand where the differences are, to better help guide the industry on what the tests look for, what the sensitivity results mean, to help give better guidance on what test to take or what does it mean with those results. So we currently have a good protocol written. We have test labs assigned. We're gathering controls, and hopefully this round robin will be actuated soon. This is one that we're going to spend a little bit of time on because the changes are great. This is one of my highlights as far as the changes coming down the line, and that's with ISO 10993-10. So Dash 10 is for sensitization and irritation. That's not going to happen for long, though, because we're actually breaking out irritation into its own standard. So ISO 10993-10 will be unique to sensitization, and the new standard Dash 23 will be for the irritation. And the reason why this is happening is because the standard is including an in vitro alternative to the irritation rabbit test. Just so you know, this is an initiative. I have to give a lot of props to Kelly Coleman from Medtronic, who has done a lot of work on this and is currently still doing a lot of work. We'll talk about it here in a minute. But we are, as an ISO committee, really looking at where we can use alternative to animal testing. And that's a great thing from animal welfare point of view, and I think that's the main reason why we're doing this. But it's also great from a company's point of view, because a lot of these in vitro tests are quicker and cheaper than the animal alternatives. So it's almost a win-win-win, and because of that, we're really trying to identify what makes sense. Still making sure we're screening for safety, because ultimately the patient is the main reason we do this, remember. But there are a lot of good science out there that is being done by lots of people to help demonstrate that these in vitro tests can be utilized in lieu of the animal tests. Particularly this irritation test that we are putting into the standard, it has to do with reconstructed human epidermis cells. And we won't go through very much detail for this just because of time, but I wanted to put them in the slides what this test looks for and how we run this test. But really what it does is, instead of putting an injection into a rabbit to look for irritation, we use this reconstructed human epidermis skin cells, and we extract a device just like we do with the test, and then we expose these skin cells to that test. We are able to use a positive and negative control, and then at the bottom of those cells we run an MTP test to, like a cytotoxicity test, to help us really evaluate the impact of these tests. For medical devices, we once again just follow ISO 10993-12. With a polar and non-polar and the same extraction parameters as in the standards. And then we put those onto these little skin cells. That's really what they are. So we have these skin cells, we have the tissue in this plate, and then we expose it to these extracts. For medical devices, we just expose them to 100 microliters of this extract, and then they're exposed for this 24 hours plus or minus 2. And then we wash off the skins and we blot them dry to make sure that they are completely clean. And then we do the procedure for the MTT test. And this is the results you're looking for. For the MTT, just so you know, if the cells are alive, they metabolize this MTT salt, which is where the MTT gets its name from, and it turns purple. So the more purple it is, the more cell viability is there. And that's what the MTT test looks for, is viability, not cell death necessarily. And so the darker the purple is, the more viable those cells are, which means the less irritation has happened. The more clear they are, they're more white, the more irritation present, because the MTT reaction did not happen. So this is kind of an overview of how the test is ran and how we analyze the data. What I want to update with you on this is right now we can run this for notified bodies. We've done this successfully. It's actually growing a lot. It's being accepted in Europe as an alternative to the animal test. The FDA right now, we're currently in a procedure with the FDA called the MDDT process, which is a way for the FDA to evaluate alternatives to these procedures and to see if they would accept it. We are currently working, Kelly Coleman from Medtronic's leading this charge, but we're currently working with the FDA, and I'm honestly not going to speak for the FDA, but the hope is that we can work through any concerns that the FDA has and get this procedure accepted by them before the end of this year. That's the hope that we have, so that we can start running this particular test for irritation, not just for Europe, but also for the U.S. markets. And once again, the benefit is that we feel like, one, we're replacing animal tests. Two, we actually think it's more predictive than the animal test. And three, it's, in the long run, going to be cheaper and quicker than the irritation. So all of that is good news, that we're getting very close to that endpoint. The next standard we're going to talk very lightly about is ISO 10993-12, and this is the one I'm most excited about, but also the one that I'm a little bit scared about as far as what's being changed. The reason why is because this is the sample preparation standard, right? So this is the one that impacts almost every single test we run, at least the ones that are extracted. And with that, there are some concerns in this standard. It hasn't been looked at for a very long time. And so we want to try to make better the sample preparation, like requiring or requesting pictures or drawings. solubility better defined. But one of the things that I really want to explore here, and I know that the committee or the working group does too, is understand better these extraction ratios. So these extraction ratios were adopted from the USP a long time ago. And a lot of them don't really have much scientific, if any, backing or evidence as far as why these extraction ratios were chosen. So do we know that 3 centimeters squared per mil is the same as the 6 centimeters squared per mil with the 60 centimeters squared and the 120 centimeters squared ratios? Do we know if 37 degrees is the same as extracting at 50 degrees is the same extracting at 72 for the different time points? There's a lot of questions as far as what these extraction ratios really mean or how they correlate with one another. And so, once again, there's some labs doing round robin to try to understand these extraction ratios better. So ultimately, there could be some changes in the historic extraction ratios, which honestly I would like, because I would like some scientific data to help support those ratios. I think that's why we're in this business, is to have some data to understand what we're doing. But on the other hand, we've run these extraction ratios for decades. So, changing them now what kind of impact would that have going forward? So we're taking it kind of seriously. I don't know if that change would happen in this particular draft, because it would, we have to understand a lot before we move. But there are some work being done right now to try to understand these extraction ratios better. We don't have a whole lot of time left, so I'm going to be kind of high level. Plus, we're going to talk more about 1718 tomorrow. So. But there are some changes to 17 and 18. So for those of you that don't know, Dash 17 has to do with a toxicological evaluation of the chemistry results. So you run the chemistry, the extractable leachables. Dash 17, some guidance on for a toxicologist on how to evaluate those endpoints. Historically, that guidance has been kind of more of a strict protocol, I guess, with particular endpoints in there as far as data goes, as far as this is the number you need to add, and it's turned more into this is how you do things, and you just kind of step-by-step procedure. And once again, we're trying to get more into allowing flexibility for scientists to make their decisions. And to be fair, Dash 17's already had that, right? I mean, it's always allowed the toxicologist to make rationale, but as soon as you put in kind of this is recommended procedure, people tend just to follow that procedure. So what we're looking at doing is allowing more flexibility by removing some of those places that called out specifics. The other thing is if you're familiar with toxicological evaluations, there's kind of this idea where if your margin of safety is over 1, you're safe. If you're below margin of safety of 1, you're unsafe. And that's kind of been just your cut dry thing. Oh, I'm going to be unsafe if I have my margin of safety under 1, but if I'm over 1, I'm fine. And really, when you look at what we do in the toxicological evaluation, there's a lot of safety factors put in there. And even in extractable leachables, there's a lot of exaggerated type of testing put into that. So just because you're under 1 doesn't necessarily mean your device is unsafe. It might mean that you've over-tested it. And over-assessed it. So it gives guidance on understanding exactly what was done and understanding on next steps on how to evaluate if you are under a margin of safety of 1. So that's, and the other part of it that I think is very useful is not all toxicologists are chemists. And so, in fact, I think in my experience, very few toxicologists are chemists. So a lot of the data the toxicologist is given, they just take it as, this is the data I need to assess. But like I mentioned before, that a lot of times that data may not be sufficient for that device based on how the chemistry is ran. So hopefully we can give more guidance on for the toxicologist to understand how applicable the extractable leachable data is to them. Now, Dash 18 is where, like I said, the new draft was just released this morning, and it's been completely rewritten a couple times. I feel I have a little soft spot for the working group, the writing committee for Dash 18, because They've been working very, very hard. Every time they get a draft, the working group puts a lot of comments in there, and then they have to go back and rewrite it. I think this is, I don't even know how many versions they're on now, but it's starting to get into a very good document now. And so we're hoping that this current draft that's out, that we have our next meeting as an ISO group in Berlin in December, and the hope is that we can talk about this current draft that's out there, some of the comments, and really get to more of a finished product. But some of the things that we're talking about is that, and Matt and Annalise will talk about this more tomorrow, but for implant devices, they really are looking for 3 polarities. So you want a polar, a non-polar, and a mid-polar. The FDA particularly really wants an exhaustive extraction, so that's being discussed into the standard. To help you understand what an exhaustive extraction is, it helps us know that we're getting at least 90% of the stuff off a material. So it's some series of testing we run before we even run the actual chemistry to know, okay, once we extract at this temperature and time, we're pretty confident we're getting at least 90% of what's there off. And the reason why that's important for a permanent implant is even if we extract at 50 degrees C for 72 hours, and even if the toxicologist takes that amount and says, oh, that's a 1-day dose for the rest of their life, there's a lot of safety built into that by itself. But the concern is what about a material that may leach, just a little bit in the first 72 hours, but then something breaks down, either a crosslinker or something in the material breaks or degrades, and then they have a bolus of material release at day 12, for example. Well, we might not know that. We wouldn't know that if we just extracted at 50 degrees C for 72 hours. So the exhaustive is meant to help us mitigate that risk for something that's in the body forever. And the last part of Dash 18 that's become very important is that these endpoints have to be addressed, especially for permanent implants, where we have to look at volatile organic compounds, semi-volatile organic compounds. The non-volatile organic compounds is really important, and metals. And I just want to, and I know they're going to talk about this more in depth tomorrow, but I just want to talk about that non-volatile. That's one endpoint that's relatively new for some companies, and it's important because there's a lot of compounds out there that are non-volatiles that would come off and could be concerning to the body. So that's one endpoint that should be addressed particularly. So the last part of Dash 18 that we're going to cover is there's some discussion still about how much quantitative data we get versus semi-quantitative data. And the reasoning behind that is when you start to set up a chemistry test, you can either really know exactly what it is, and certain compounds, so be very specific, I'm looking for DHP, and when we do that, we can do that really well and really understand how much DHP is in the material. Or we can just screen for anything that we can possibly detect. But when you do that, you lose some of that specificity when you look at a single compound, and you get some semi-quantitative data. So when is quantitative data needed versus semi-quantitative data? Because when is an actual identified compound needed versus when is a screening method more appropriate? So that's some discussion that we're talking about in this current draft. Making sure we have that correct identification like I talked about. With this, you can always set up the perfect chemistry approach, which is screen for everything that's there, go back and validate every single compound to get the quantitative amount. No one would argue that that's the best scientific approach. But we have to balance the cost of this work versus the cost of an in vivo testing, because ultimately we want to be able to make sure people are safe, but also want to help replace some of this animal work with this chemistry. So that's the discussion we're having all there, is how deep do we make this chemistry to where it starts becoming cost prohibitive for device manufacturers. Here's just some of the information for you as far as the in vitro irritation and sensitization in vitro development where we're at, some white papers that you can go back on, and we talked a lot about the FDA guidance document, and that's a link for that. Now we'll turn the time over to Chris to go into questions and answers.
All right. Well, thank you, Thor, for a great presentation. And now, before we begin with today's Q&A, please direct your attention to our webinar survey available on the right-hand side of the presentation window. If you close the survey, you can reopen the widget by clicking on the icon along the bottom of your screen. Thank you in advance for filling out the feedback form. Your participation in this survey allows us to better serve you. We're now going to move on to the question and answer portion of our event. As a reminder, to participate in our Q&A session, just type your question into the text box located to the right of the presentation window, or click on the Q&A icon at the bottom of your screen. And if we're not able to get to your question today, someone will get back to you after the program is over. So let's get started with our first question. Our first question for you, Thor, if your device passes a cytotoxicity, will it also pass irritation tests? In other words, can a device flunk the irritation test but pass on cytotox?
That's a great question. And so there has been some work done internally at Nelson Laboratories where we've compared. So basically what we did is went back, I think, 10 years, and we looked at every time a device failed and what test did it fail. And I think it was about 93% of the time, if a device fails, it's cytotoxicity. So it's safe to say it's the most sensitive test. Now, there are a lot of times where something is cytotoxic and it's also potential irritant. The one that comes off the top of my head is the detergent, for example. A detergent, residual detergent, a lot of times will kill cells, and that detergent is a possible irritant too. So there are times where you could fail a cytotox test, and that would be a symbol or a precursor to, yes, I would fail an irritation. But one of the downfalls of cytotoxicity is we can't really use a true nonpolar extract, and I'm not going to get too technical into it, but because of the cells, we can really only use a polar with a little bit of nonpolar sprinkled in. And what that means is that if your compound on your device is lipophilic, then that means it would come off in oils more. It's possible that it wouldn't come off the device in an MEM test, but then we put it into cottonseed oil, for example, and inject that into a rabbit, or put onto the in vitro irritation test, and it would come off into that solution and be at high enough amounts to cause an irritation. So there's not a great answer. A lot of times, cytotox is a great precursor, a screening test to screen for the other tests, but it's not a fail-safe, right? There are times that we'll see an irritation. There are times we'll see acute systemic toxicity, where it did not fail a cytotox test. And that's about 8% of the time, I believe, where we see that. So I hope that answered the question.
All right. Well, thank you, Thor. We got another question here for you that says, the standard states that failure modes are out of the scope. So how do we address the risks?
Yeah, very good question. And this comes more from a regulatory standpoint, where if the regulators see a possible failure, the best one I have is like a balloon, for example, inside the body, where the balloon, not a lot of times, but it's known that the balloons could burst, and so the interior of that balloon would be exposed to the body where it's not intended to. And so what I do when I write a risk assessment for a device is I first have to understand the risk, and I use 14971 a lot for this, is what's the likelihood of that risk to happen? Is my tip of this device going to break off during surgery and be left behind? If that's a likely thing to happen, then I should address that risk. I should look at implantation. I should look at the impact of leaving that device behind. Or if the balloon's going to pop, I need to address those materials inside. My personal view is I would take those risks separately, because I would not want to test them together, because if there's something bad inside the balloon, for example, I don't want that to ruin everything else. And so if you do have a failure in the risk part, you want to understand the real risk of that failure in the test with the failure of the device. So that's the approach we want to do. We first want to understand, is it a true risk? Is it likely to happen in a clinical setting? And if it is, you treat it just like you would any other biocompatibility risk.
All right. Thank you, Thor. Another question here for you asks, can we use supplier information to know what the chemical composition is, or can only extractables give us that chemical information?
You know, I've seen a lot of this happening in the industry. So what will happen is the manufacturers are starting to push the need for leachables and extractables down to their suppliers. So basically they're saying, hey, I need to know material information per the new standard. So you need to do an extractable leachable test and give me everything you see and the quantities that you see off of your material. And then they can use that, a supplier or a manufacturer could use that in their assessment. But remember, biocompatibility is just not about materials, it's about processing. So that's only half of what you do to it. You then have to understand all the processing, including sterilization, what could impact those leachables. So just because your supplier says, oh, I've got this set of leachables coming off my device, you then have to understand how does your processing could change that leachability of that material.
Thank you, Thor. Another question here for you says, what should you do if you have a material that is proprietary to your vendor?
Yeah, this happens all the time. So that's the hard part about doing a material assessment is right now getting that information. I just did a presentation in Lyon in July with a bunch of raw material manufacturers, and they were really concerned about this new standard because they were already feeling it. They were feeling a lot of pressure as far as giving formulations and things to their customers and saying this is proprietary. I don't want to hand this over. So the hard part of this is if your supplier, you can only do so much for the supplier. My recommendation always is to try to get that from them when you're trying to develop a deal, because that tends to be easier when you're trying to get business than when you've already have a relationship with them. But if you're not getting proprietary information, that you just have to now do more yourself. So that requires you to do more extractable leachable testing yourself, because you're not looking to reverse formulate that material necessarily. What you're trying to understand is what comes off it from a leachable and extractable point of view, and you can do that yourself.
All right, Thor, we have another one for you here that says, if the E&L test results are satisfactory, for example, showing a good margin of safety, how do you determine which biocompatibility tests do not need to be done?
Yeah, so there's two answers to this. I think that the FDA particularly has come out in their guidance document to mention genotoxicity and subacute subchronic toxicity, well, all systemic toxicities, as the endpoints that they will allow chemistry margins of safety to be used for. The key there, though, and this is the second part of the question, is it really depends on what data is available in the literature for what endpoints. For some compounds that have been studied forever, we can find systemic toxicity endpoints, we can find genotoxicity endpoints, we can find carcinogenicity endpoints, and all of those then can be utilized to address that material. But if a material we can only find genotoxicity, for example, then we don't know if systemic toxicity would have a lower kind of amount that would be toxic. So in that point, we may need to go back and do some systemic toxicity. The true answer, though, is most of the time compounds have good genotoxicity, good systemic toxicity, and carcinogenicity information. So those three endpoints are the ones that are most commonly justified out of.
All right, thank you, Thor. Another question here for you says, what is your interpretation of indirect contact in regard to contact with surgical gloves, but not in the same sense of the biocomp definition?
Yeah, so with surgical gloves, a lot of times, so you really want to look at indirect contact. So my definition of indirect contact from the biocompatibility is does gas or water or some other kind of material fluid pass through the device into the body, like an IV bag would be indirect contact. With surgical gloves, some people would talk about, I've heard on the phone call, well, the physician could touch the distal end of my device with the surgical glove, and then the device goes into the body. Historically, that has not been looked at as indirect contact. Now, if that surgical glove could touch the patient, then obviously it's direct contact. Or if it would be in the contact with a lot of fluids that then go back into the patient, then that would be indirect patient contacting. And just remember that the FDA in their guidance document and in our newest ISO 10993, we talk about practitioner contact. So if the practitioner, so like the surgeon or the nurse, contacts your device without personal protective equipment. If like a glove touches their bare skin, that glove also has to assess the risk to the practitioner.
All right, thank you, Thor. A lot of questions coming in. Our next one here for you is, what is the relevance of these tests, in vitro cytotoxicity, to inhalation devices?
Yeah, so that opens up a whole other realm. So there's a whole 18562 is an inhalation, the standard for inhalation biocompatibility. And what we tried to do in that standard is really try to point out what tests are unique to that type of contact. So I'm not saying cytotoxicity doesn't have a risk, because sometimes those gas path devices would have skin contact or mucosal membrane contact too, which then would have those risks involved. But if you're just looking at a gas path and no other contact with the body, then you'd be more likely to look at what's the impact of particulates being shed, or what's the chemistry impact. So what could be leached from, because gas is acting like a liquid, so what could be pulled from the material from that gas and delivered to the body. So that's more of the risk for that type of contact. But just because you have gas contact, there's also some devices that would then also have skin and mucosal contact.
All right, thank you, Thor. Another question here for you says, is exhaustive or exaggerated extraction and identification of extracts mandatory in all cases?
No. So it's really only being utilized right now with chemistry. There has been some push in genotoxicity, but once again, chemistry may be replacing genotox if you do it correctly. The other tests like sensitization, irritation, we don't really evaluate exhaustive, or the exaggerated is there, right? So when you extract at 50 degrees C for 72 hours, that's an exaggerated extraction, and that is being pushed by the FDA and notified bodies. But the exhaustive extraction is mostly only used for chemistry for implant or for devices that have longer than 72-hour contact with the body. So if you have a device that only has 48 hours of contact, then we want to exaggerate that extraction. But if you have a permanent contacting device, that's when an exhaustive is required.
All right, thank you, Thor. Looks like we have time for maybe one or two more. The next one here says, with regard to the ISO 10993-1 version that just came out, is it effective immediately, or is there an effective date?
Yeah, great question. So I'm going to answer this kind of in a weird way. So it is effective immediately, but what happens when these standards are released? So ISO releases it, and then the governmental agencies will take, like ANSI, for example, will then take it and adopt it. So there will be an ANSI ISO or BSI ISO or EN ISO. So because this is new, the ISO just came out, and so now it goes into those individual areas to be adopted. So that still is happening. So you may not see like your notified body, for example, going to it yet until they're like BSI or something like that will or European Union will adopt it, but it's coming quickly.
All right, we have time for just one more quick one here. It says, what will be the consequence if companies continue to checkbox instead of risk-based approaches?
Yeah, so we see this already in Europe. So the notified bodies, when they checkbox and they do not do a risk assessment up front, we've seen the notified bodies actually reject that and say there was not a risk assessment done, even if they did all the testing. The FDA right now still kind of does they still allow kind of the checkbox mentality, because for them it's a risk assessment unless the material is somehow concerning to them. So if there's either the material or the intended use of the device is a unique risk for them, then they'll push back for like nano or maybe some type of unique administration to the body. But I actually see that changing. I think the more and more the FDA starts to look at this risk-based approach, the more and more they look at pre-subs to work with the FDA, I see them going more and more requiring the risk assessment. So the FDA already requires a biological evaluation report at the end, which Audrey will talk about on Thursday. But the notified bodies are definitely pushing it, rejecting submissions that don't have a risk assessment. And I see the FDA really maybe not rejecting, but pushing those risk assessments moving forward.
All right. Well, thank you, Thor, for a great presentation and a great Q&A session. That is all the time that we do have for questions for today. If we didn't get to your question, again, someone will get back to you after the program is over. Within the next 24 hours, you'll receive a personalized follow-up email with details and a link to today's presentation on demand. Please feel free to invite your colleagues and peers who may not have been available to listen to today's event. This webinar is copyright 2018 by UBM. The presentation materials are owned by or copyright by Nelson Laboratories, and the individual speaker is solely responsible for his content and opinions. On behalf of Thor Rollins, I'm Chris Keach. We'd like to thank you for attending today's course, and be sure to join us tomorrow and Thursday.